The excretory system in human beings consists of a pair of kidneys. A magnified view of the longitudinal section of kidney shows two prominent regions, a cortex and a medulla. As we zoom into the cortex region, it shows the presence of numerous nephrons. Each nephron has a Bowman's capsule, a proximal convoluted tubule, a loop of Henle, a distal convoluted tubule that leads to a collecting duct. Nephrons are convoluted in the cortex region and straighten out as loops in the medulla region. Each nephron has a Bowman's capsule, which is a cup-shaped structure that has a layer made of squamous epithelial cells. In the cavity of the Bowman's capsule, there is a bundle of capillaries known as the glomerulus. The blood vessel entering the Bowman's capsule is known as the efferent arteriole and is thicker than the blood vessel exiting the capsule, which is thinner and is known as the efferent arteriole. The Bowman's capsule and the glomerulus together form the Malpighian body. This capsule extends into the proximal convoluted tubule (PCT) on the inner side. This tubule is lined by a brush-bordered epithelium with numerous microvilli. This PCT leads into the loop of Henle, which lies in the medulla region. It is U-shaped and has a descending limb and an ascending limb. This further leads into the distal convoluted tubule, which is again seen in the cortex region, and it finally joins the collecting duct. As blood flows through the glomerulus, water, salts, and waste diffuse into the Bowman's capsule. This liquid then passes into the PCT and the loop of Henle, where water and important salts are absorbed back into the blood vessel. Only the waste material like urea and some water molecules proceed into the DCT, where more water may get absorbed into the interstitial fluid. This liquid formed is the urine, which is released into the collecting duct.
Traveling through the nephron. The proximal tubule initiates at the Bowman's capsule. 65% of filtrate moves from the proximal tubule back into the blood. This process is called reabsorption. The movement of water occurs secondary to that of sodium and other osmolytes. The movement of sodium from the tubular fluid into the cell is mediated by a battery of sodium-coupled exchanges, which move sodium and hydrogen in opposite directions across the cell wall. Consequently, the reabsorption of sodium largely occurs in exchange for the secretion of hydrogen into the filtrate. Water reabsorption occurs through the water channels aquaporin 1 and 7. As the nephron leaves the cortex and descends into the renal medulla, the reabsorption of water and sodium are separated. This happens because the ascending and descending limbs of Henle have different permeabilities for water and sodium. The descending limb of the loop of Henle has a relatively low permeability to sodium but a high permeability to water due to the presence of aquaporin 1 channels. In the ascending limb, sodium, potassium and chloride are transported from the luminal fluid into the cell via the cotransporter in a ratio of 1 to 1 to 2. 5 to 10% of sodium reabsorption occurs in the distal connecting tubule through the sodium channels ENaC and NCC. This region does not have aquaporin water channels and therefore its permeability to water is low. Here, sodium leaves the filtrate but water does not. The collecting duct is responsible for the fine-tuning of sodium reabsorption. The movement of water and sodium from the lumen to the blood are under the regulation of vasopressin and aldosterone, which are released depending on whether the body needs to conserve water and sodium.
This finally trickles through the kidney into the ureter, which helps the passage of urine into the urinary bladder. After a while, the urine can be excreted from the bladder.